रोहित जी, डॉक्टर साहब ने ज्वाइन कर लिया है, आपको होस्ट बनाइए उनको, डॉक्टर सी भारद्वाज साहब। ओके सर। डॉक्टर साहब नमस्कार, जय श्री राम।
जय श्री राम, नमस्कार। सेम
डॉक्टर
पर्सन विद
साहब,
a new
विद इन
topic.
विद इन 5 मिनट्स स्टार्ट करते हैं।
Okay, sir. I'm online. I'll
जी
just
जी जी, हाँ जी सर, फैकल्टी और साइंटिस्ट ज्वाइन कर रहे हैं। 250 प्लस जैसी स्ट्रेंथ हो जाती है वैसे
sure,
स्टार्ट
sir.
करते हैं।
Sure, sir. Sure.
रोहित जी, डॉक्टर प्रगति श्रीवास्तव को होस्ट बना दीजिए। नंदिनी, आप सुन पा रही हैं मुझे?
हाँ जी सर।
ऑन एन एवरेज 220 प्लस पार्टिसिपेंट्स जूम पर है, लगभग 200 प्लस यूट्यूब पे भी ज्वाइन करे हैं। तो 400-450 पार्टिसिपेंट्स ने ज्वाइन कर लिया है। ठीक है, तो रेस्ट ऑफ द पार्टिसिपेंट्स हैं वो इन बिटवीन भी ज्वाइन कर सकते हैं। तो हम ये कह सकते हैं कि 400 प्लस स्ट्रेंथ हमारे पास आ गई है। तो
हाँ
आप
जी सर।
मॉडरेटिंग स्टार्ट कर सकते हैं विद इन 30 सेकेंड में। ठीक है।
Good evening, everyone, all the respected dignitaries, distinguished speakers, mentors, and participants from India and abroad. Welcome to the day 21 of International Winter School Bharat 2025, a platform dedicated to advancing resilient innovation-led agriculture. I am Nandini Sharma, Assistant Director HR, AgriMeet Foundation Bharat, and it is pleasure to host today's session highlighting leadership, institution building, and scalable solutions for sustainable agriculture. The Winter School, jointly led by AgriMeet Foundation Bharat and UGC MMTTC Jodhpur, with ICR and university partners, which fosters collaboration across research, education, and extension. It emphasizes climate resilience, value chains, and technology deployment for farmer impact. Our theme, Boosting Holistic Agriculture Through Research, Awareness, and Training, reflects the collective mission to convert science into solutions across region and value system. So today I am very much honored and very much excited to introduce our profound honor to welcome an extraordinary scientist, transformational leader, and one of the most respected plant breeding experts in our country, Dr. Chellapilla Bhardwaj, Principal Scientist, Division of Genetics, ICR, IR in New Delhi. With a distinguished career spanning more than 3 decades, Dr. Bhardwaj has contributed immensely to soybean genetics, chickpea improvement, pulse research, and modern plant breeding technologies. His work beautifully connects laboratory innovations with field level solutions, benefiting farmers, researchers, students, and the nation at large. Sir's brilliance has been recognized with some of the most prestigious awards: the Gregor Mendel Memorial Award 2025, Professor M.S. Swaminathan Award 2023, Best Scientist Award 2023, and numerous international recognitions as well. Globally, he represents India with unmatched dignity, serving as adjunct professor at the University of Western Australia, adjunct associate professor at Murdoch University. These rare international appointments reflect the global value of his scientific leadership. Beyond his research accomplishments, Dr. Bharadwaj is a phenomenal teacher and a mentor. He has guided over 49 research scholars, shaping future generations of plant breeder and biotechnologists. His students describe him as a man of clarity, discipline, compassion, and scientific excellence. Ladies and gentlemen, when we speak of the future of Indian agriculture, name like Dr. Chellapilla Bhardwaj sir stand tall. Scientists who push boundaries, build capacities, and translate research into real impact. Sir, your presence today elevates day 21 of UGC ICR AMF Winter School Bharat 2025 to a moment of great pride and learning. On the behalf of entire organizing committee, we welcome you with immense respect and gratitude. We are very much grateful for your presence. We eagerly look forward to your words of wisdom. Over to you. The session is yours, sir. Thank you so much.
Thank you so much, Nandini. Thank you, Ankurji, it was always a pleasure speaking on the forum. The specialty of this forum is it cuts across students who are the future leaders and across the people who are at helm of affairs in policy to that in active research, and therefore it builds nation, and Bharat 2025 is very apt. With that, I hope you have permitted me to share my screen.
Sir, please, please.
Uh, I don't know why I'm unable to share my screen. Okay, window, entire screen. Why it's not coming?
Rohit ji, admin Mahoday.
No, sir. No, sir. Sir, you click on entire screen, na? Just click on share button, like the blue button. After clicking on entire screen, there is a button for share.
Oh,
Yeah, yeah, yeah. It's working.
Done,
Yes,
sir, done, sir.
done, sir, done.
It's perfectly visible.
Yes, sir. In PPT mode, sir, can you do like hide? Then go for PPT mode. Yes. And
Done, sir.
can
Done,
yes, sir.
done, sir.
Sir, you are muted, sir. Badao, sir.
रोहित क्या हुआ महाराज?
take a second.
Sir,
Let me. I have stopped share. Now you are able to hear me.
Yes, sir. Yes, sir.
yes, sir. Yes, sir.
Let me share screen. I click on this. Yes, sir. You are able to hear me too.
Yes, sir.
Yes,
Yes,
sir.
sir.
आप भी
Yes, sir.
हाँ आप देख पा रहे हैं सर बिल्कुल.
Okay. So, what I would be like, I would be liking to speak is basically how artificial intelligence, which is coming in a big way, the big data would be applied in plant breeding and trade discovery. Already a large amount of data and what you call as review articles are there, but basically I'll be speaking from my own labs experience and the work that we have carried out through my students and collaborators. Now, whenever we speak of artificial intelligence and machine learning, we need to understand some of the patterns. Like, we do have some of the models which make computation, which is basically a neural network feasible. This becomes a subset of the machine. That means these heuristic models with large amount of data sets garnered over samples and over areas and over time tend to make the machine learn to give better predictions. As better predictions increase, today artificial intelligence is basically a synergistic, that is, it collates different papers or different data sets that are available to it, either in the cloud to which it has an access, and it generates. Therefore, it is predictive in nature but not mostly exact. Therefore, whenever you use even AI to write a para, you get a line that there can be mistakes as it is predictive based on data available. Now, this is very dangerous phenomenon. Like, for example, it is like a colony of ants where all the ants have three legs. And over a long period of years, one ant is born which has four legs. Now, what happens is the mother, the father is very worried. They take it to the grandfather, says, "Let us take it to the king, because this ant has more appendages, just for the story I'm saying, has four legs." So, all the ministers sit together, and then what they do? They remove one appendage, and now this ant, which was otherwise normal, also does 1 2 3 hop. And as of current, that's what is happening with artificial intelligence, because it is collating things. Beyond that, it is still has to be trained or it has to be learnt upon by the particular machine. Machine here is the model that is able to better predict. And how it is going to better predict? When it has more and more heuristic, but the field data. If the field data is totally wrong, it will predict a wrong prediction. So the most important thing in a machine learning model is what are the decision trees, or what are the random forest model, what is the k nearest neighbor model or k means. Therefore, your multi-layer perception and encoders are able to better bring out this particular subsets. So you have deep, as I said, a machine learning or a deep learning or natural language processing. I will not go much deeper into it, but speak of how AI would come in agriculture. But in case of science, large number of data sets are coming over the minute. For example, each lab is working on developing transcriptomics, because somewhere from 10 lakhs for a single sample, we have come down to 25K. So large amount of data set analyses are going, and a huge amount of decision is able to be done only based on what you call as what are the inferences that you are putting, what are the what we call as the conditions of modeling that we are putting. But it is very highly limited, because what we speak when we speak of artificial intelligence is how a machine is going to simulate human intelligence, which includes, as of this stage, it is learning, but then it has to make a reason and also make a self correction. The simple machine learning model is a correlation model. Now, whenever we speak of correlations, we as a human brain have a set of data because of our practical experience that we have sent along, and a machine has a data which is actually in that. Now, for example, for a breeder, let us say we have bold seeded chickpea lines and which are high yielding, and we have one or two, or let's say 10% of these subset is of small seeded lines which are lower in yield. Now you make a correlation, a simple p is equal to sigma x plus ai, then it will give that the bolder the seed, the higher the yield, which may not be the case, because correlation is always per se for that set, and the moment the set changes, the correlations change. But the human perception clearly tells the more the grains, the smaller the size, but the yield is higher. So whenever you get such what we call as missense correlations, I don't call them nonsense, you need to look where we are going wrong. But the whole role of AI in agriculture is how do you enhance productivity? How do you manage the large amount of phenotypic, high throughput phenotypic, high throughput genotypic data and make meaningful decisions for breeding or for crop improvement? And how do you actually correlate multilinear? Till now, we were going at single linear models. How do you correlate multilinear models so that the model itself is sustainable and does better predictions? We have a large amount of tools. If you're looking at phenotypic, you have the R tools, or they have the TableU or NumPy. If you're looking at imaging, we have just started earlier with C++ and Python, and then we started moving ahead with Sentinel and GE tools. But then the most important is what are the decision support tools which are inferred from these models, maybe CropX or farm logs. But what it basically helps us, and these are the four aspects which I would be covering with respect to the work that we have done. How do you make selections in a large populations, and how these selections can be better when compared to phenotypic selections? How can you do trait mapping using artificial intelligence? What are the different prediction models that you can operate on or that we have operated on, and what are the different modeling techniques. Now you need to carefully please keep one thing in mind. I am presenting from what we have done in lab. Huge amount of work is done, particularly in case of rice, wheat, and maize. But being chickpea, my area of field and my authority with my collaborators, for example, in selections with Ikrisat, with Varshne, Manish Rurkiwal, Nicholas from UK, and the group. So we have developed the strategies how you can uh, predict genomic information using breeding programs, not only for just India, but for Asia and Africa. How these predictions can be maximized and how can you optimize chickpea yield, for example, using biophysical and machine learning tools. Dr. Arun Singh, Premila Krishnan, and our group has worked. Similarly, how can you make trait mapping, for example, high throughput phenotyping technologies, how do they help in making better selections? Or how do you model and what are the different models that we had employed and which is best for genome enabled prediction models for yield traits in chickpea? But before I start my program, I should amply thank all my co-authors, Dr. Neeraj Kumar, my colleague at IRI who had given the previous lecture, Manish Rurkiwal, who was at ICRISAT, later moved to Khalifa Center of Engineering and Biotechnology, and now is leading Reliance Industries on as an agri research lead. Professor Rajiv K. Varshne, who is professor at Murdoch University. Snehapriya Papul Reddy, who is now working at University of Western Australia. Sudhir K, my close friend who helps in all high throughput phenotyping at National Nanaji Deshmukh Phenomic Center at IRI. Singh Aran and Anita Krishnan P, and for the first time, even before other crops, chickpea could do image based predictions. Thanks, that was a great work. And our close friends, Patil from Dharwad, who assists in almost all our works from phenotyping to genotyping, and my two colleagues, Jyoti and Darshan, who are working with me and have tried to compile this whole synthesis of this lecture from the various publications that have brought out from this lab. Now, if you tell that if all your friends jumped into the well, will you? As I said, what happens? The kid will use his drawn intelligence and will say no. And the machine will say yes because majority did, like the sheep. And this is a very important aspect because machine does analytical learning whereas humans do predictive learning. The best example is when a child is born, how does it recognize that its mother has to breastfeed or bottle feed him or her? The thing is, the child gargles and mother kisses. The child cries and mother slowly smooths her cheeks and hair. But when the child hits her legs and makes gurgling and crying voice, that's the time when the mother actually feeds. So the first impression is like in the Paul Ligar's 8 caste model, it happens with acquisition, interest, desire, conviction, action, and satisfaction. This is how human prediction models develop from a young child. And that is why we say that family is the most important aspect in molding the human brain. But a machine learning just takes the predictive information from already done aspects. And now this is another peculiar confrontation between the AI researchers and the breeders like me, because AI researchers only predict, but it is basically breeders who have to use this. So what happens is AI researchers may work like a bulb. They predict that how much of height, how much of intensity, how much of this thing, and breeders need to do it. So now what's happening is there needs to be a marriage, and this is very, very important. You need to understand that in 1900s, Before 1900s, all our selections were based on what we call as an art by the breeder's capacity to identify. And the best selections earlier were made by Babylonians and Assyrians when they're selecting the date palm. There was no comparison, there was no significance. It is good, it is good. Now, when Sir Ronald A. Fisher has introduced biometrical genetics for the first time, it has become very, very important that we need to understand that the biometrics or measuring genetics through various models had come into what we call as agricultural research. Slowly, what happened around 1970s with Carey Mullis identifying and bringing out the polymerase chain reaction machine or the PCR, that now the genomics guys started slowly coming in, and it almost took 20 to 30 years for the marriage between the genomic researchers and the breeders. Then what happened basically was now slowly data scientists are coming who are trying to evaluate the various models, bring in machine learning tools, and this is how that it is going to affect now. With large amount of data that is available, now people are trying to look how artificial intelligence tools will be thinking of how it will match the natural stupidity in a way, because researcher is basically a madman if you look into a common man's language. Why artificial intelligence and plant breeding? We have a large examples of artificial intelligence that play a major role in plant breeding. Artificial intelligence, for example, would come in a large way in case of genomic selections, because you have large data points. Which of the data points to select? What are the models which will tell if these data points are selected, it will give the best combination for a yield? And which of the data points to be combined, that infers that which are the plants which are calling these data points. These data points are basically genomic points, and then you need to cross them so that you can have a better round of selection. Or how you will predict environmental data, and so that that environmental data better predicts the phenotypic data. Or how are you going to make selections at the target production environment sitting at somewhere else? And this is what is going to happen in the future. A scientist who is sitting in USA can predict based on good, if but all this requires solid and very good phenotypic data. Based on the phenotypic data, and you can see the CGIAR, the NARS systems have developed a large amount of data since 1950s. There is a huge amount of data on rice, this thing. And you have target production environment. India is an excellent example where zoning has been done. So how you develop and utilize this large amount of phenotypic data? Maybe you have some historic lines and the current lines whom you can genotype and understand what is the basic group of genes that are giving high yield, and those rare genes that give the extra effort in terms of stabilizing a field, in terms of what we call as disease resistance, what we call as nutritional quality. And all these is not new, but this is there in our what we call as breeding value. Earlier, breeding value took into consideration multiple phenotypic data, the year data, and the deviation from environment and the deviation of the genotype from the environment to predict the breeding value of a genotype. What is breeding value? In simple sense, it says the performance of the progeny to that of its parental population as derived from base population. And all breeders try to increase their breeding value as we go from generation to generation. If not, what you will be doing? Taking the best developed line that is developed at IIPR Kanpur and actually taking the best developed line that is developed at IRI, You cross them, you'll create a stabilizing normal curve. But what I want to move, the mean of my normal curve, I would like to move it towards yield. Maybe I would like to move the mean with lower days to flowering and maturity. That is a early genotype with higher yield. So what are the genotypes I have to predict to actually do? There are many good statistical models, what we call as multilinear models. You have blubs and blues that have come. But when genomics had come, when you got lower, very low genotyping costs, so you can genotype a large amount of populations and then look which individuals carry it. So this is like a basic genetic gain acceleration perform. Means earlier we had these entries in the gene banks, and then what we tried to do based on our phenotypic data, we used to make the crosses. Maybe we used to develop the core, because as a breeder, I will not be able to actually use all the lines. So I used to use the basic diverse set, and then we always try to make some crosses and then evaluate these in a diallele or test cross fashion, or develop them as a SST and then evaluate, and the best predicted lines we used to cross again, and then again evaluate the progenies, and then the base seed we used to cross, and we used to develop either populations or increase the breeding value per se in case of self-pollinated crops. But then basically a technique of speed breeding where we can take 3 or 4 generations. Hickey is 6 generations, and I can say that even 3 generations plus 3 generations in 2 years, 6 generations by simple breeding at IRI, or let's say Northern India, Central India, and Dharwad, over 2 years will get you 6 generations in 2 years. But then the problem is you don't know why you are forwarding, because in this such a type of shuttle, super shuttle breeding, I will be not be able to select for the phenotypes that I desire unless I go into field and evaluate, which is not so I'm doing in a polyhouse and in a different environment. So what I do, I actually identify the base pool of the genotypes based on their genotyping data and the allelic data that has been developed for various genes, and then I make a selection. Mind you, that these are putative selectants, but sure, they carry the haplotype or the most desired haplotype for our genes or our what we call as alleles in consideration, and we cross them. So the second generation, again in an artificial, either in a polyhouse or in a fast breeding shuttle situation, or if you're using speed breeding like PAU has developed, like ICRISAT has developed, like IRA is developing, and IAPR is developing, and so is ICARDA. So what we are trying to do when you're taking 6 generations, I pick out from almost 200 plants or 300 plants seedling stage, I cull out and keep just those few plants that carry the genes. Now, since there are few plants, even at off-season nurseries, I can make, or at F6 where I put into PYT and then evaluate them, and then the best ones I create. So this becomes first cycle, or you may actually look at yields and go till station trials and advanced trials and do there. That becomes later generation genomic selection. But what as a breeder we aim is, when we are doing earlier, you try to have a cycle of genomic selection where you try to increase therefore the number of plants. From a large number of plants that you are carrying, almost 200, 300, you handle large number of crosses and you bring down to a small set which can easily be handled. Now, if there was Abhishek, he would have said, "Ah, wait, Bharadwaj, there are different designs. You needn't test all these of your F6 from these crosses that you put together at every place. There are spatial partial designs. So I can test few of the genotypes with common checks at each place, and then I can accumulate the data and also better predict." So better prediction model with panel selection and having a large amount of sequencing and multiomics data, as well as having data from space, that is what we are going to speak of, using either sensors, that's what Sneha has done, or high throughput phenotyping techniques, that's what Sudhir is doing, that you can actually better predict in those lines and then actually go for developing the varieties. Now, of course, epistasis and genome editing is coming in a big way to have greater genetic gains. But friends, what I would like to put along this course that has come is from panel selection to the biological big data to phenotypic prediction to the selector lines. You have a huge amount of data and huge amount of innovations that have come in case of biological data management. We had computer heuristic models that used to develop, and then we put them to the human data. For example, in a store, how many will purchase, let's say, Lux soap, and that's how the backend used to go. Now, exactly we are trying to do based on the phenotypic yield data. So, whenever you are looking at characterization of germplasm, for example, machine learning for genetic diversity, it looks into enhancing genetic diversity, maintaining dynamic genetic variability, or taking out traits which are essential for adaptation. There are traits which are essential for adaptation and fecundity, and there are traits which are essential for breeder for yield. And please let us know these two traits are different. So you need to look how genetic diversity needs to be maintained for a germplasm person. Now we have a geo-referenced data utilization, which integrates the total bioclimatic data and soil gradient data, or identifies genomic signatures for crop adaptability. Now you may think this may be just for breeders. No, it is very, very important for even companies who are giving insurance, who make coefficients. Maybe it is a newer thing into the Indian thing, but where you have a large farms and large yields, like Australia, where 200 hectares is minimum size, like USA, where 800 hectares. For example, if you are looking at the middle of USA, where you have whirlwinds common, or to the western part where tornadoes are common, you know which season is common over almost 20-30 year data, and they could predict it. Or in case of fires in the most important valley of USA, they do predict that this is the season of fire. And mind you, they haven't issued insurances to these houses for fire in the recent fire case. So these predictive data are very important. Now, sitting here, you can predict the target environment simulations based on your phenotypic, but you require to have very good phenotypic data. And we need to understand that the ACRIPS in the Indian system are sitting on a wealth of information with this phenotypic data, and we require a good bioinformatics to analyze this. In case of chickpea, 40 years phenotypic data. In case of rice, 80 years of phenotypic data, zone wise or a state wise, and come out with those predictable variables, which if bred into a variety can give a super variety. And that's what we have done in case of Pusa chickpea mana, which is called as a super chickpea variety. Or that's what I was saying, ki how do you integrate the artificial intelligence driven genomic prediction to that of target environment simulations, so that you can come with a set of predictable variables that a bioinformatics otherwise calls as the superior haplotypes, whose information is passed to the breeders. Now, whenever you're looking at conventional breeding, so what I do basically, which I was speaking before, is that you have a germplasm pool with target population environments. You evaluate with multi-environment trials. You pick out the best lines. You constitute a base, or what we call as a predictive population. And then this small predictive population is again given to the NARS partners. The best lines are picked up and crossed together to have a breeding value more than the baseline. So it is based on performance. and based on phenotypic data analysis. Now, with a large amount of genotyping, and I would call you it as envirotyping, maybe I'm throwing out newer words. Envirotyping is high throughput phenotyping, both spatial as well as physical. You have, and you correlate that with some models to the genotyping data. So you have a genomic prediction models, and based on these models, you select the genotype, you cross, get better genomic estimated breeding value. And as you will see in my next few slides, how better is the GEBV to that of PBV, phenotypic breeding values. So you have a training population. This training population, why you require a training population? Because you require a standardized data on which a machine can predict. Once a machine is predicted, you need to test whether the machine has done or not, and you need to make the machine learn. At some stages, we are quite lucky of our ACRIP system, but in some models where you're starting for the first time, maybe dry root rot, we need to start a training population and look how this goes. Maybe salinity, because as my colleague Sanwal says, it's not just sodium chloride salinity, Dr. Saab. आप लोग जो बायोटेक्नोलॉजी वाले करते हैं, this is wrong. It is a multi-salt salinity that works in soils, unlike in pots or in your hydroponics. He's perfectly right. And then the most important is, how do you cross-validate these models? At 80% training, at 20% training, how many times you have validated this model? Just 2 times in 2 environments? Did you validate in 5 times, that is 5 environments? Or like Icarda or Icrisat, you have done in multiple environments across Asia and Africa and the so on. How much of input data you are having? Are you using still SSRs for phenotyping? Have you gone to SNPs? Or you have, how much of SNP data you're generating? You're generating a 50K SNP data, you're generating a 100K SNP data. Are you looking at mid-density platforms which are giving sufficient information and bringing down the costs? And exactly how better it gives for your output data. So there have been large amounts of innovations in phenotyping, as you see. For example, if you are looking at plant phenomics, from that of simple handheld meters, we have gone down to the image-based meters, to the rail-based platforms. We have developed a good LizzieScan facility, thanks to Professor Kadambot Siddiq, Professor Varshne, and the group. Some people use phenocards, or are you using no artificial drones? That needs to be actually looked into. Or are you using sensor based and developing a large amount of data? So you have a large amount of high throughput phenotyping data. You have a large amount of genomic data. So what you are going to do? The application part is very, very huge, from plant stress biology, where it forecasts a large amount of genomic crossovers, looking at genomic editing at DNA methylation product, or you're predicting genetic gene promoters and regulatory elements which play a major role, and how these elements, when you're using and crossing, are moving. But more important is always the gene works through metabolic networks. So are you identifying tissue specific gene variations? Why this is becoming important? Already Rajiv and his group at Murdo are working at spatial transcriptomics, single cell transcriptomics. Because if you look at a guard cell, which is around the pore, or at the pore cells, which is around the micro root hairs, the same pore is used for entering of the rhizobium. The same pore is used for the entering of fusarium. What does the guard cell do? What it secretes, or when a rhizobium comes, what it helps to allow it to symbiotically enter, and in a resistant line, what does that guards, what is the pore cell around the pore does, or what genes are activated and what metabolic elements they release that fusarium is unable to enter into a resistant and a tolerant line. This is very, very important. So earlier, just looking at just root transcriptomics, you need to look at specific genes that are activated at the point of entries. Maybe leaf guards, maybe at the cutting points, you need to look. Then most important is we have errors both at phenotyping, we have errors both at sequencing, we have certain outthrowers which are outliers. Maybe you need to identify why it is an outlier. Maybe it is a highly resistant line. Maybe in that genotype it is giving the best genomic predictions in form of yield, in form of tillers, in form of number of pods. And then most important is you identify that subset of elements which are maximum present in two genotypes so that you can cross them. There are many challenges in artificial intelligence based predictions. Friends, we are just learning. Now, for example, we have just a single binary vision. And if you look at the left side windmill, it appears as a single windmill and having 6 wheels. The moment you develop a binary vision which gives you depth, you do understand that these are two windmills, not a single one. So it becomes very, very important that how you are. For example, if a machine is asked to make clusters, it will just either make it either on the color, or if this thing is fed, or on spicy, right? So every candy has a specific color. It is either reddish or bluish. So if the red candy is red, the maroon candy is red, the purple candy is red, the orange candy is red. It could not distinguish at this stage because hue is not fed into it. You have three basic things, isn't it? So hue is one that actually distinguish the shades of a color. So it clusters all of them into red. It's perfectly right. It clusters all of them into blue because of its predictive nature. So you have just an algorithm which does not know relationship between these four colors, which are different shades of red, and it doesn't distinguish between these four shapes of color, which has different shades of blue. So given this set of features, unlike humans, machine has only two subsets. But as a human, I conclude that there are 8 clusters, not just two. And for me, these 8 clusters are perfect, that they vary from red, maroon, pink, flamingo, blue, turquoise, seaweed, and ocean. And for me, each is different. If I ask my better half, she'll say, "What nail polish you got? I asked you to get flamingo, and you got me a red one. You don't know this?" Because males have mostly binary colors. Oh, just a joke. Similarly, no, this is a peculiar case again from how machine algorithm predicts. This is a wolf. Machine predicted as a wolf. Beautiful. This is a husky. Machine predicted as a husky. This is a husky. Machine predicted it as a husky. This is a wolf. Machine predicted as a wolf. Now, if this was a predicted as a wolf, but it was actually a husky. Now, if you look into this of 10 cases, 9 cases it has done perfect. So 90% prediction. Wow, 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 very good. No, friends, please, you need to revisit your models. Machine has neither predicted husky or it neither predicted wolf. What it did, it predicted snow. It classified snow or no snow. And wherever snow is there, what basically happened? It predicted as wolf. You just look into any wolf picture and you will see there is a snow. Wherever there was no snow, it predicted as husky, right? Because there was snow around, it predicted a wolf, but actually it was a husky. Because there was no snow, it predicted husky, and it was actually a wolf. So the machine learning learned that wherever there is snow, it is a wolf. So it classified snow or no snow. It didn't classify. So this is a false recognition, or it's a false positive. And we need to be very, very careful when we are making such predictions. So, how do you overcome this? You actually try to overcome by having a large amount of data sets, and these large amount of data sets are innumerably large to have better prediction. So it is just like that, that where there was snow it was wolf, and where there was no snow it was husky. But since there was snow here, it predicted this as basically husky. It removed husky, it never recognized husky, it recognized snow or no snow. So it is very important how we interpret our models and how do we go ahead. So, what are the AI applications in phenotyping for precision breeding? So, you can imagine that from just 10x capabilities of breeding pipelines, just by using simple SSRs, if you can increase by 2x, we can increase by almost 8x by superpooling. That means 800 times you can increase the size and still work out with simple machine learning models and modern phenotyping and genotyping techniques. So if you are using almost 8 years, then you just require 3 years, and a poor breeder like me who has just a greenhouse and collaborations with ICRISAT can do only in 3 or 6 years. But whatever it is, so we moved away from plant breeding 1.0, that is 99 to 2004, to that of 2 to that of plant breeding 3. That means global and faster evaluation of data, with developing of genotyping data, developing more accurate phenotyping and genotyping data, to making more accurate digitally enabled predicted data that a breeder can actually use. So you have a pipeline, and every private company works as a pipeline. So what is happening now is, earlier a plant breeder was a good person who could select based on art. Later, because of the biometrical tools, he could select better because he could evaluate. Now, based on earlier based SSRs based phenotyping, we could select much better. But present on the genomic tools, now from selection, we are now moving to designing. So designing requires generating of data, both phenotypic and genotypic, with both scale and accuracy. Now, not only that, can you fast place the products? And that is the market. But at the same time, can you optimize a product for a specific environment? For example, the Chhattisgarh requires short duration, heat and drought tolerant lines. So what are the lines? Hey Manish, can you tell me? And he says, yes, doctor, these are the lines, these are the genes that are required, or this is the haplotype that is required. This haplotype is because all the germplasm is now being characterized at genomic level, as being done at ICRISAT and at the group of partners together in the mission mode program, in case of chickpea, in case of wheat, in case of uh, uh, other millet crops. So now you have a whole, so you can blast this what you call as the best haplotype and identify which are the plants that are carrying them, and then you can cross. But maybe because this is pre-breeding, because basically they may not be, you need to bring out the best yield based haplotype for your traits. Therefore, AI enabled high throughput phenotyping increases the accuracy of decisions. AI allows us to understand the performance of material in any grower's field. The innovations in phenotyping and models maximize the potential of genetics. Earlier, just 4 testing locations, you see, you have 4 data points. Now, based on sparse designs, you can grow across 10 or 20, and you have 1 million data points, both from spatial data, high throughput phenotyping data, and genotyping data. Now, because data collection on large scale is much cheaper, if I run my one chickpea genotype plant through the LISI scan of Dr. Mahesh and Dr. Sudhir, they will give me 1 million phenotypic data points. RN Sync with Dawn and Premila can also generate a huge amount of qualitative data, be it UV sensor data, and then you can test them at more diverse conditions. And we have used all these 3 in chickpea. As I was speaking of genomic selection and breeding value, this was what I was saying. From a simple 10-year model, if marker assisted breeding could bring you to a 6-year model, the genomic selection as well as rapid generation advancement or double haploid technique can bring a release of a variety to a lower model, but at the same time, a breeder can handle large populations. So that's what basically genomic selection does. And since Neeraj has touched with it, I will just use it as a tool to make you understand that all these are high yielding cows. Let's say this is, of course, they resemble here the same cow, but let us say from this parent 1 and parent 2, you had a large generation of cows, and basically all plant breeding techniques have come from the animal breeding techniques, which have come from human genome analysis. Now, you need to understand that there are some good genes in this, which is for higher yield and let us say greater milking period. Which of these cows and the ox carry these genes? We have a phenotyping data. Now we have a genotyping data, and you have a large amount of SNPs that has made. What we do, this is our training population. So we basically evaluate them and see if our phenotypic data and the genotypic data on which you made predictions, basically these are associations matched. So you create a cycle which helps in lining. And that's what exactly we did at IRI with our close friend, Professor Rajiv Varshne, Dr. Manish Rurkiwal, Anuj Tikneni, that we developed a huge amount of elite breeding lines, which we made as a training population. Now understand that training population is not a germplasm set, and we evaluated it over locations, over environments, over 3 seasons. So we had 6 huge amount of data points. We started with DAT sequencing. We faced problems in the model predictions, and then we moved to GBS, and now we moved to Axiom SNP array of chickpea, which is Rajiv's groups and is with Dharmo. And we restructured our training population using all the advanced genotypes that came in ACREP, and then could make greater genomic evaluations. Now, it doesn't stop at there. Whether it is basically fitting, and there are numerous ways. For example, someone wants to go to Kedarnath, he can walk, he can go by a pony, someone can carry him, or he can go by a helicopter. But which one suits our model best? For us, for example, I don't have money enough, then I may have to walk. But I have enough money and I want to make best selection, which saves money, which saves time and everything. Maybe I'll go through an agent with a helicopter. So we have used different models, as I indicated before: ridge regression, kinship Gauss, Bayes, lasso, random forest. And we saw that with random forest, we could get best prediction accuracies, even for yield traits under drought situations, like drought susceptibility index, seed yield. Of course, you get even single predictions. But now the most important was our prediction accuracy varied from 0.89 to 91. And if we have a zero point 0.7 breeding value prediction accuracy, that's what you do. In heritability and genetic advance, you immediately pick that trait and say that you need to select the parents based on these traits. And we have moved further, that we have checked whether this prediction accuracy is common across groups, and we have seen that Kabuli less predicted Desi except for seed size, but Desi better predicted Kabuli. Then we said, oops, there is a genetic divergence, and that's what our 3000 sequencing paper, which had come in Nature, has proved. So what we have done is we have said, ki, let Kabuli predict Kabuli and Desi predict Desi, and we didn't stop at that. When Dr. Manish and Varshne were predicting using genomic selection, I tried to make 3000 single plant selections based on the phenotype using the plant architecture: number of pods per plant, number of seeds per pod, number of primary branches, number of secondary branches. And then when we started doing that, generally in case of breeding, we go for multivariate prediction models. And before we have published this paper, even in genomic selection, they were all multivariate prediction models. But what we started doing is looking at multivariate prediction models. We have taken 3 or 4 variates using the genomic selection. And when we compared between univariate and multivariate, the accuracy was perfect, and the multivariate models were either predicting better or predicting equal with the univariate genomic models. Now the question had come, can we look at whether the genomic models can predict with same accuracy that we breeders do? Basically, all of us are breeders. I am a breeder. Most of my colleagues, Dr. Neeraj, if you're looking, we are all breeders, and we wanted to look whether the genomic prediction model is giving same as the phenomic prediction models. And when we look at for either for plant height, or you're looking at days to flowering, days to maturity, 100 seed weight, seed yield per plant, the percentage prediction was good. And if you are looking at predicted, the yellow line, for example, if you are looking at the black one, which is genomic predicted models, it was doing better. You look at 100 seed weight, look at plant height, you look at days to maturity, you look at seed yield per plant, it was predicting better. Days to flowering, shorter durations, you look and you find that the genomic prediction models were actually predicting much better compared to our phenotypic prediction models, which gave our strength that the model we have chosen for our set is good. Now the problem is the model has to be specifically chosen. The model, for example, you have developed for integrated pest management for Heliothis cannot fit for aphids. You need to develop a model. This is where, as I said, visual selection has better predicted or predicted with equal but better efficiency, because we could make it in off seasons. We haven't tested for 6 generations these plants and then selected at PYT, and we almost have 100 lines now, because we have a problem of just entering 2 lines per year. We have a huge amount of lines that are backstacked with us, which give better prediction. So now we have started deployment of these populations into National Chickpea Program. One more thing within the next 2-3 slides, which I will actually deal and conclude, is how spatial expressions can be used for disease or yield predictions. Now this is our field, the Wilczek plot, which is spread over 2.5 to 2.8 acres of land. Which has been well coordinated in IRA experimental fields. So what we did is we have the field observations, and we wanted to see if the observations are taken by a drone, can we make some prediction experiments? And we have taken almost at 4 different growth stages. We have uniform checks both the susceptible and the tolerant lines. We had the F4 data, which tells which group of lines has what level of resistance. We have taken the 8 biophysical matters, parameters of field, 3 thermal indices, and 45 visible indices. We fed them to machine and we started looking at what is the estimated yield. Of course, as I said, in our models, where we had to calibrate these multilinear models to how do we predict chickpea yield at 85 days, where you know that around 35 days the early wilting occurs in case of chickpea in the north west plain zones, and flowering stages one where peak wilting occurs. So what we have done is we had these pictures, but we looked at 95 to 90, 85 to 90 days thermal scannings. We tried to validate these models. So when we started looking at the 85 to 90 days models, we found that the GLM and XGB were the best models in calibration as well as validation. And then we started looking how this calibration, validation at all stages, and actually it affects. So what we tried to do is what is the impact of various levels of chickpea on crop biophysical parameters to predict the yield in multilinear models. The machine learning multilinear models employed in our study could effectively predict. Now, when we look into things, we find that when you are using the values of R square or D index or RMSE, they have better predicted. Of course, for each parameter, the improvement can be attributed because we just didn't go by the hyperspectral images or the above spectral images. We have included, as I said, almost 32 parameters, which included 8 yield traits, which included almost about 21 physiological traits, and then more than 130, which we have dropped down to bring out into a subset of the hyperspectral images. So, as I said, we were closely linking plant health with all the biophysical models, and we could develop a model that predicts, in case of wilt in northwest plain zone, how yield will be affected. Maybe this can be used for frost predictions. It needs to be worked into. Now, as we have seen, that cubist and random forest tree based models like XGB, we could find that these are better performing, but XGB outperformed because it has taken a machine learning multilinear models. One more work that we have done, I think this is the last slide, is that how high throughput phenotyping for terminal drought stress. It is an open access paper with plant stress. My student Sneha and Sudhir have worked a lot with the UWA team. So we had images that were acquired across the models. Now we checked out how we could generate this hyperspectral images at the pot level and look at our data. So when we started looking at yield, growth, and photosynthetic traits, mind you, again, these are about 20 or 30, including the root images of the marker-assisted bread lines for drought and the donor ICC 4958 and the recurrent parents, we could get a significant correlations. Now this will help me a grade that our MABC lines can be pumped only at the terminal, not at the terminal stages, but somewhere at the flowering stage, at the pre-flowering stage, and I can predict which of the lines will have a better yield prediction. It may carry the genes, and those actually I can multiply. So what I was saying is genotype data is tabular. So you need to not have a tabular data, you need to have a multi-dimensional data and a pipeline, both bioinformatic pipeline and the breeding pipeline, but with clear focused goals that will help you. If your data is not big, then just 2 or 3 people, as I said, you can have false correlations and associations. The scaling laws always say that the data is to be large. Even when we are calculating our significant at 5%, it is not that you do 100 times and you get 5. Wrong, as per the hypothesis. You do infinity number of times, and in that you can go wrong in 5%. No, miss, scaling errors can occur. So you have a large type of models. I think we don't have much time over it. But as I said, if the domain is high throughput phenotyping, we need to look at some of the already well worked out models. But our problems are to how to reduce manual laboring efforts, how to improve the accuracy of measurements. If you are looking at integration of multi-omic data, you need to establish a roadmap or a pipeline. That's what we were doing in the Bill and Melinda Gates, now Gates Foundation program, where we were developing pipelines and almost 7 crops with 200 researchers working. If you are looking at identifying the most different variant, so what are these deep variant calling tools? Because that variant may be for carrying genes for wilt tolerance. It may not be for high yield, but this becomes important when you are looking at germplasm characterization. If you are looking at functional discoveries, so you need to understand what are the candidate genes which are responsible from already available databases. What are the biological processes that are carrying? Because the candidate genes are not single for most of the abiotic stresses. You will have a group of genes. Now you need to identify which of the genotype is having most of these genes. That means, or the allelic variant which it is having, and then select the genotype. So you have different tools that have been worked. So you have a large amount of, again, variation models. And this is from Rajiv's latest paper, how artificial intelligence can guide fast forward breeding for accelerated genetic guides. So you have a large amount of phenotyping, high throughput phenotyping, and high throughput sequencing data. You need to identify these blocks which are there, which actually carry it, and then check out. Suppose I carry cross this haplotype 1 with haplotype 2. What is the prediction that I get? I really develop a population using speed breeding, selecting for the superior combined haplotypes. And with that, I go for real phenotyping, saving on cost and labor. So, what is the big key that I'm going to say when I'm saying, speaking of fast forward breeding in this thing, is that it is collaborations of the physiologist, of the pathologist, of the genomic person to that of the breeder, more collaborations with the economist, because it is finally the market oriented breeding that is going to fetch. I may have a line 100% resistant to fusarium, but the thing is, it doesn't yield even 10 pods per plant. What's the use? And it is enabled to cross with most because its combining ability is very poor. What's the use? So you need to look at that case some special techniques, and these in short will help in developing climate resilient crops to withstand environmental stresses. They will help to optimize agricultural practices for sustainable and future demand. Why I'm putting this here is, now certain times I needn't reinvent what you call as research. Now, because of collaborations with CGIAR, ICAR, and NARS, now you know that if Madhya Pradesh is going to become very hot, then you have a large amount of data from Nandyal, Gulbarga, Latur. You also have from Africa. So you now have already phenotypes what Dr. Jaylakshmi is looking at, or Dr. Anita Babbar is looking at for short duration, more number of primary branches, and the genes that Raji would indicate having the haplotype. And if, let's say, tomorrow the duration is too short over 30 years, then I'm ready with genotypes. So predictive breeding can be done. You can enhance the efficiency and the innumerable solutions that you can expand for a crop breeding potential. So this becomes important. So you need to understand that the future belongs to nation with grains and not guns. I add to it that it also belongs to the person who is able to analyze the data. If not, it is another bioinformatic phenotyping. If not, it is molecular phenotyping. You sit on a large pile of data but don't know what to do. So I hope me, the coming generations, and the young people like you can carry the father of Green Revolution India, Professor M.S. Swaminathan sir's legacy forward. Now, I would like to thank that this is not a work of me, but I represent not only this team, all my project associates, staff, Apurva, Ravpreet, Uttarayan, huge number of other students too, Bharat, Kapil, the collaborations at the national level, whether it was National Institute of Plant Biotechnology, IAPR, IRI, 21 divisions, NIPGR, thank you Swaroop, thank you Praveen, Jagdish and the group. Thank you, Dr. Preeti from Kota, and similarly, Dr. Anita Babbar Madam and Jayalakshmi from Angrao, the whole lot of Murdoch team, Professor Rajiv Varshne, Professor Anu Chitikaneni, and all friends. The CIMET group, Abhishek Rathor, Yesh and whole. The University of Western Australia and its partners, Professor Kadambod Siddiq, Harvey, Sneha, and the group. ICRISAT, Rakesh, a close guru bhai and a close friend, where we fight, we discuss, we laugh, and we pray together. Dr. Shiv Kumar, my student Tapan, Preeti, and Neha from ICARDA. I am really thankful, but this would not have been possible but for my funders from DBT, Gates Foundation, IRI, ICAR, the SERP projects. And I thank you, Ankur, that you have brought about and given me a chance to speak to a large audience on this upcoming role of artificial intelligence. It has come in many other things with respect to plant breeding, genotype development, as well as predictive breeding. I think I should close, I should stop share, and then goodbye. Welcome back. Thank you.
इस बेहतरीन लेक्चर में यदि पार्टिसिपेंट्स की, हमारे साइंटिस्ट जो फैकल्टी स्टूडेंट्स ज्वाइन किए हैं, कोई उनका कोई संदेह है, कोई डाउट्स है, अगर वो इससे रिलेटेड कोई बात करना चाहते हैं डॉक्टर साहब से, तो अपने आप को अनम्यूट करके 1 to 1 बात कर सकते हैं. चैट बॉक्स में सराहना मिलने लगी है सर.
मैं वही ध्यान है नहीं है, मेरे क्वेश्चंस के लिए वेट कर रहा हूँ. फ्रेंड्स, आई एम थैंकफुल, बट as many questions you ask, I also learn. Maybe I'm not an expert at everything, but we'll try to answer, or we'll try to get back through Ankurji.
जो पार्टिसिपेंट्स हैं वो 1 to 1 सर से बात कर सकते हैं. सर हिंदी में भी कंफर्टेबल हैं, इंग्लिश में तो कंफर्टेबल हैं ही, है ना, तो कोई
तेलुगु
दिक्कत
में भी
नहीं.
है.
हाँ, तेलुगु भी आती है सर को थोड़ी थोड़ी.
तमिल थोड़ी तो नहीं, तेलुगु तो मेरा मातृभाषा है.
अच्छा, मात्र.
And what all I presented, again I'm putting, is the work that we have done at our lab. Much more and huge amount of work is now being done at Murdo and the UWA, and me and Rakesh are trying to bring about a good amount of projects. Yes, please.
Hello, sir.
Arjun ji, bataiye.
Sir, my question is, can I use AI and its technology for For screening of large number of mutants when we go for mutation breeding in
For nutrition
hybrids.
ऐसा है ना, first you need to identify the model. You understand? The model is a जरिया which will tell you to pick up. अब आप पूछ रहे हो कि मुझे ट्रेन में जाना है, कहाँ जाना है, तो न्यूट्रिशन के लिए जाना है. वेरी गुड. बट इतनी लार्ज पॉपुलेशन को सही प्रेडिक्शन करने के लिए आपको एक मॉडल को ट्रेन करना ही पड़ेगा. क्यों करना पड़ेगा? हर एक मॉडल की रिक्वायरमेंट्स, हर एक पॉपुलेशन की रिक्वायरमेंट्स अलग है. मैं वही बोला, कैंसर की दवा मलेरिया के लिए यूज नहीं कर सकते. तो आपको कैंसर की दवा डेवलप करने के लिए एक लार्ज अमाउंट ऑफ मॉडल पे उन्होंने टेस्ट किया है, सेफ्टी टेस्ट. इसी तरह आपको एक पॉपुलेशन, बेस पॉपुलेशन बनानी पड़ेगी जिसे हम ट्रेनिंग सेट कहते हैं. And that training set is specifically different for a situation. Nahin to what happens? You'll be doing a large amount of molecular phenotyping data, molecular data. You'll develop a large amount of genomic data, and it doesn't predict. Basically, all these models, either by machine or by biometrics, work with association and correlation. So you should know if I drop a pen and you know that in a minute, and that pen takes 1 minute to fall down from a terrace. And in India, when a pen is dropped, in a minute there are 3 children that are born. And now you predict that Dr. Bharadwaj drops a pen, and if it is taking 3 minutes, 3 children are born. So you will feed the model wrong. So you need to first develop a model, you need to validate a model, and then you can use for predictions, right? So what I'm saying is you require collaborations. पहले भी था. I don't know why slowly breeders got into silos. If you look, any variety that is developed from IRI has the pathologist, has the physiologist, has the biochemist, has the agronomist names. We don't work in silos. And that's why I think when Neeraj was presenting, we said we don't have boundaries in our research, therefore we had gates in our fields. We work together with the NARS. We not only handhold, we learn together with NARS. And therefore, I'm not asking you develop, but you need to identify collaborative partners who can train a model and give to Arjun, and therefore he can use it, right? But you can certainly use.
Okay.
Sir, is there any available model for
There are many available models, Pete, but you need to look, or your bioinformatics needs to look. For example, you are working in, let's say, Pigeon P, which model better fits for yield and nutrition prediction? There are large number of models, and I don't say every model fits everywhere. Like, for example, when we are doing genomic selection, random forest model better predicted. But when we were doing spatial things, what Dr. Arun Singh and Premila have done, it is GLM models that have predicted. Both the clauses were different. So you need to, every single model does not fit at every place.
Okay, sir. Thank you, sir.
Hello, sir. Good evening.
1 second. Amrita already have indicated the paper is an open access paper. You can take my email ID and I can send the paper, or you can type Ram, Premila, chickpea, wilt, spatial model, you will get it. Yes, madam, I'm sorry.
Yeah. Hello, sir. Good evening, Andy. Okay, I'm from Hyderabad, and I have one question, sir. What do you see as the biggest breakthroughs coming in AI-driven plant breeding over the next 5 years?
Madam,
Can
please, I'm sorry.
I'm just continue, sir. So AI can it help us identifying the nutrition efficiency varieties that require fewer fertilizers, or what we say that climate resilient seeds? Can it be helpful in the next 5 years? Can we see this change?
Madam, AI, just like biometrics, is a data analytical tool. Please understand this. For this data, it requires a large amount of data to be generated and fed to it so that it makes perfect model. Now, as an example, if I take more number of, we are working on which crop?
We are working
Rice.
on multiple crops, Sandy.
It's okay. Let
All the
us take
farmers.
rice.
Yeah.
Let us take rice. है
Yeah.
ना?
Yeah.
Now, we look at that the South Indian rice varieties are superior, right? Let us say we are
Right.
with this conclusion. Most of the South Indian rice varieties, living the sannalu or the things which consumers consume are bold seeded. L by B ratio is high. Or let's say you've taken land races as your base population and L by B is high, which are also higher in protein, then the model will predict it has bigger L by B ratio is related to protein. Now, these are just two variables. Now you add the temperature. Now you look at fertility data. So you require a multimodal data. And let me tell you, it's a prediction tool, right?
Right.
It's a prediction tool. So it will predict from your base population. You need to train that model to predict it better. Once you do it, yes, if you have a targeted trait for which you have developed a population, you can make faster selections without evaluating for the trait. Now let us look. We have developed, we are in the process, I don't say, in case of chickpea, identifying high protein, what we call as haplotypes. Dr. Neeraj, my colleague, is working on that. Now he has identified haplotype. We have a base population. We are working together with Dr. Yogesh. This year we'll be initiating the crosses.
Okay.
Now our idea is I needn't go for the lab analysis from the initial because we are developing a speed breeding facility. So we think that within the next 6 or 4 generations that I can easily take up, I am able to select lines based on these prediction models. I validate them in basic growing in the next year where I will take up 4 generations. So in F5, you do early generation selection in case of nutritional traits. You do late generation selection in case of yield traits. That's why F4, F5 family character starts and then you go till F6 or F7 for yield analysis. But for nutritional traits which are governed by larger chunk of genes, if so, then we start evaluating F4. But at F4 or F5, now I can have fewer lines which are having higher protein.
Understood. Yeah. Got it.
But the whole thing depends on, for example, let's say you're from Andhra Pradesh, I'm from Andhra Pradesh, and we are working on, I'm sorry, you're Telugu speaking and I'm Telugu
Yeah.
speaking, and we are working on nutrition.
Right.
When we have only Ankur who is working on heat, we say ki Telugu speaking people work on nutrition. That is
Yeah.
an association. So that's what the AI model has
आप
done.
लोग अगर दोनों लोग तेलुगु जानते हो तो 2 मिनट तेलुगु में बात कर लीजिए, मुझे भी सुनने को मिलेगा कि
अरे
तेलुगु होता
नहीं,
क्या
ऐसा
है।
नहीं, सभी वक्ताएं हैं यहाँ।
तेलुगु में
मैं-
एक्चुअली हम लोग अलग से बात कर लेंगे अंकर जी, वो प्रॉब्लम नहीं है। वैसे भी मिलना भी है सर से भी। एंड सर, इवन आई वांटेड टू कनेक्ट बिकॉज वी आर वर्किंग विद द फार्मर्स एंड आई हैव नियरली 2 lakh farmers working connected with us, and we are working on soil and nutrition of the produce as well. So I would definitely need your help and support on that.
Sure.
So I'll connect through अंकर जी। अंकर
Sure.
जी, आपका हेल्प तो चाहिए यहाँ पर भी फिर से।
बिल्कुल मैडम, एनी टाइम।
Right, sir.
Manjunath ji, you said you're working on your doctoral research for different colored maize. Maize is well worked out, and Krasa from Summit is working on AI, right? But literally at this stage, is the anthocyanin color changing from sweet corn stage? No idea because I'm not a maize breeder. But what I say is selection is market oriented breeding, and basically the color change is not in predictive breeding but directive breeding. For example, for those people who are from Bihar, they prefer white typed corns. But those people who are into poultry require hard corns which are yellow in nature with more anthocyanin content. So these are totally different. I would request you to connect to Dr. Firoz Hussain, who can better guide you in this.
Next, please. नंदिनी, आप हैं?
Avinash has asked what is the future role of AI
हाँ जी
in
सर.
seed science.
जी.
A last question. Okay, Avinash, I think we are going a lot back when you compare with private companies. Do you know that each seed is RA tagged in case of a private company in the research? And sitting there, when a guy takes his parameter multispectral analysis tool to that plant, the data directly goes in case of Corteva to their central unit. So the data is directly fed. So you can imagine we are lacking forward. We still in the national system are going by what Bharadwaj thinks is best. We are not driven by market. Many people are coming. We are driven by farmer, but somewhere we forget that farmer is driven not only by yield but also market. Now the simple example is, for example, Kabuli. Bold seed size fetches him 12,000. Now today's market rate is about 15,000 a quintal, to that of Desi chickpea, which is 3,000 to 5,000. Let's say 3,000. The Desi chickpea yields about 2.5 tons, 25. 25 threes are 75. And the Kabuli chickpea yields 2 tons, 20 into 15,000. So straight away, the market oriented. You need to look, for example, the color as you have put. The market oriented breeder in Corteva tells ki, okay, small sized soybeans are required in case of China, in case of Japan, where they go for fermented soybean products. So higher protein is required. Whereas Indian market requires larger seed size, because the larger is the seed size, the greater is the oil content. So you need to look at the market oriented demand. So this is a pipeline. The market oriented breeder tells, okay, this is the requirement, this is the disease, this is the thing. Immediately the pipeline pushes to germplasm. The germplasm guys say, hey, I have these 3 lines which are having the 3 characters that you have said. Then immediately biotechnology guy comes. He says, okay, these 3 lines, okay, these are the outlier genes. If not, he maps it and he identifies what are the genes available. And then these are pushed. Okay, these are the lines which are carrying these genes and having this character. And then you now cross it. Now, when you crossed it, the bioinformatics guy will be tracking you the flow of the gene in the foreground and the yield related genes in the background, what we call as foreground and background selection. Putting it very, very simple, Ankarji. It's not as simple as that. And then finally, you have lines, a fewer lines that are having genes for, let's say, in case of chickpea, wilt resistance. Shaila has shown me ascochyta blight resistance and yield. And then you have a set of lines, very few lines, which you can test over larger locations. This is the thing. Fine.
Nandini.
हाँ जी सर.
Concluding remark.
Thank you so
दीजिए.
much, sir. We are very grateful, and your lecture today has truly deepened our understanding of modern plant breeding, pulse improvement, and the future direction of genetic research in India. The clarity with which you explained complex concepts, your practical wisdom, and your decades of experience have given all of us a new perspective. We are especially grateful for the way you connected scientific innovation with field level impact. Your guidance, your vision, and your commitment to empowering researchers and farmers align, inspire everyone. Us. Sir, it is honor for the UGC ICR AgriMeet Foundation Winter School Bharat 2025 to host a scientist of your status. We express our heartfelt gratitude for taking out your valuable time for us, for sharing your knowledge so generously, and for motivating our participants throughout your words. On the behalf of entire organizing committee and all the participants, thank you so once again for truly
Thank you.
remarkable session. Thank you so much.
Jai Shri Ram. Namaskar.
गुरु जी आपको मेरा प्रणाम, बहुत बहुत धन्यवाद। जय
Thank
श्री
you,
राम।
sir. Jai
जी,
Shri. Namaskar.
सभी पार्टिसिपेंट्स लीव कर सकते हैं। सबको गुड नाइट,
Shubh Ratri.
शुभ रात्रि जी।